coronary circulation? We do so at the base of the aorta. So since we started this course, we've always talked about these big blood vessels that come off the arch of the aorta, and a lot of people think about them as being the first major blood vessels that come off, but they're not. Actually, right at the beginning of the ascending aorta, in fact, so close to the left ventricle that when the semilunar valve flaps open, they actually block the entrances to these blood vessels, is the beginning of the coronary circulation. So it's very, very close to the left ventricle. So we're coming out of the left ventricle, we go past the aortic semilunar valve flaps, and right at the base of the aorta, ascending aorta, right there, the first branches that are going to come off is going to be these coronary arteries. We have the right coronary artery come off and the left coronary artery come off. The right then divides into the marginal and the posterior interventricular artery. The left coronary artery comes off and gives you the anterior interventricular artery and the circumflex artery. These interventricular arteries are sitting right on the interventricular septum, the wall that divides the two ventricles, so they help to actually delineate which side is which. So if we look at this picture, we can see these different blood vessels. And so here is your ascending aorta. So the beginning of the ascending aorta, right here, is the opening into these blood vessels. Here's the right coronary artery, and that right coronary artery is going to come around and give you the marginal here, and then the posterior interventricular artery, while the left coronary artery is going to come around and give you the anterior interventricular and the circumflex, which you see there. The nice thing is, as you notice, there's a lot of anastomoses between these. For example, near the apex of the heart, there's an anastomosis between the anterior and the posterior interventricular arteries. And so a lot of parts of the walls will have this, these anastomoses that are going to serve different parts, which ends up being very important if there's some sort of blockage. We then drain back through veins. You can see the different veins here, which all end up draining into the coronary sinus, which we know is on the back side of the heart, just below the left atrium. But it's going to head over towards the right side and dump in that deoxygenated blood into the right side. If we do end up with an area that does not get appropriate blood flow, we can end up with a myocardial infarction or a heart attack, and it's a result of the obstruction or blockage of a coronary artery. The problem is, and if you remember back to histology, is that these cells of the wall, heart wall, these cardiac muscle cells are no longer undergo mitosis, and so if you kill off a bunch of these cells, what actually ends up being put back in their place is going to be scar tissue, and so that's a non-contractile, and so that can cause problems. Well, as I mentioned, the entrances into these two arteries are so close to the beginning of the base of the aorta that when the semilunar valves open, the aortic semilunar valves open, they actually block the entrances into these two different coronary arteries. So there's a point where you get maximal blood flow into the myocardium, the muscle of the heart, and that occurs when the heart's relaxed. There's very little blood flow going through the coronary circulation when the heart is contracting. Two reasons why, if you think about it. First is the compression of the coronary arteries due to the contraction of the myocardium. These blood vessels are traveling through the heart wall. If the heart wall is contracting, it's going to close down a lot of these blood vessels while they're contracting, and so you have to wait till the muscles relax to allow the blood to go through these different vessels. The other point is that when the ventricles are contracting and they've gotten enough pressure to push the blood out the outflow tract, the semilunar valves actually block the entrances into the coronary circulation. And so you have to wait till the heart relaxes, the blood comes back towards the heart, closes the semilunar valves, and now the blood can go into the coronary circulation. Questions?
Is there any significance to that?
I don't know, it's kind of a really weird setup if you think about it. I don't know if other animals have the same setup or not. I haven't looked at that. Interesting. part hopefully should sound familiar. Cardiac muscle of the heart is really cool in the fact that some of it's able to depolarize on its own without any input from the nervous system. And so when we look at the heart wall and we look at the cardiac muscle, 99% of it is contractile cardiac muscle. But the other 1% of those cardiac muscle cells are non-contractile and are called cardiac conducting cells or autorhythmic cells because they are going to be self-excitable, meaning they're able to depolarize on their own without any input from the nervous system to get to threshold and generate an action potentials. These non-contractile cardiac cells organize into nodes and highly specialized conducting fibers called Purkinje fibers, and they're responsible for initiating the signal for contraction and also distributing that signal throughout the heart wall. The rate of the cardiac intrinsic muscle contraction is set by these pacemaker cells, but can be then altered by the nervous system. Sympathetic division, the autonomic nervous system is going to speed these guys up. Parasympathetic is going to decelerate them. We also see things like hormones can impact intrinsic rates as well. So this is the what we looked at before in histology is going to be the pathway that we're going to see this signal going through the heart. And what we see is that the sinoatrial node, interventricular node, the atrioventricular bundle or bundle of His, right and left bundle branches, Purkinje fibers, are all made up of these autorhythmic cells. Everybody's trying to depolarize on their own to threshold, but it's these guys that get there first. And because they get there first, they're the ones in charge. And left on their own, the SA node would like to depolarize on its own 100 times per minute. But remember, the autonomic nervous system's coming in, and at rest, the parasympathetic division's in charge, and it's going to slow them down to about 70 or 75 beats per minute. Okay? But if you would cut those nerves coming into the heart, it would speed up to 100 beats per minute. If you're looking for the SA node is located inferior to the superior vena cava in the superior aspect of the right atrium. And so it's going to be in this area right here. So these get to threshold first, generate an action potential, which you can see here. That signal is going to spread through gap junctions to all of the cells of the atria that actually do the contraction. There's their signal to contract. That signal also goes to the internodal pathway to the atrioventricular node, where the signal is held for 0.1 second to give time for the atria to contract before the ventricles get the signal to contract, because you want the atria to contract, then the ventricles. Get that last 30% of blood down into the ventricles, now let the ventricles contract. Signal goes to the AV node, now it's going to go through its only electrical signal down to the ventricles. There's no connection electronically or electrically between the atria and the ventricles except for this bundle of His. There are no gap junctions between these two areas. So if you would cut that bundle of His, the ventricles would never get the signal to go ahead and do the contraction. But our signal is going to go through the bundle of His into the right and left bundle branches down to the apex and then up the sides to most of the contractile cells, including the papillary muscles that are going to take up the slack in the chordae tendineae and hold on to the atrioventricular valve flaps. Remember the reason for this distribution, this pattern of distribution. Because think about it, the outflow tracts are up here. Think of it as a tube of toothpaste. If you want to get that blood up and out, if you want to get that toothpaste up and out, you always start at the closed end and work your way towards the open end. So now our signal is going to come up and out, and our blood does the same thing. If you didn't like that picture, this does the same thing as well. Anybody have any questions?